Hello everybody, so my name is Matt. I'm a final year PhD student in the top and Thalassanos labs at the ISMB in London. I'm going to be talking to you today about our work devising a scoring function for monolinks from a cross-linking experiment. So for those of you who aren't familiar with XLMS, it is a technique to generate solvent-accessible distance restraints for structural modeling purposes. So we'll first of all take our protein in solution, and then we'll react it with a crosslinker. So today I'm talking entirely about BS3 or DSS. These are most popular types of crosslinkers. They're homobifunctional, containing two N-hydroxysuccinimide groups, which have primary reactivity with lysines in the N-termini, but also side reactivity with serines, threonines, and tyrosines. So first of all, it will react with solvent-accessible residues on the protein surface that are of the correct type. And if there's another residue of the correct type, which is also on the protein surface and close to it, then it may link those two residues together. And that will yield four different, when we digest the protein, it will yield four different types of molecules. We have our crosslinks, which are the most information-rich type of molecule, giving us residue exposure information for two residues, as well as distance information between the two. We have our loop links, which are similar, but both residues linked together on the same peptide, so this distance information is slightly less useful. We then have our monolinks. Now these are different in that only one end of the crosslinker has actually been reacted with. Therefore, we have no distance information, but we do have residue exposure information. Now, the reason we want to use these more is because although they are less informationally useful, there are far more of them in an XLMS dataset. And currently, researchers don't really use them in the modeling of protein structures. So we want to devise a method to use them. So the next step is to remove as many of these unmodified peptides as possible using peptide enrichment, and then we'll perform LC-MS and MES analysis and then data analysis to identify the residues that have been modified with a crosslinker. So our aims were to design a scoring function for scoring model structures using monolinks. We want to combine that with our previously defined M and XL score that uses crosslinks, and then we want to test these scoring functions. So to score model structures based on monolinks, I created the Monolink depth score, or MODS. This score uses a parameter called residue depth, which can be calculated as the distance of a residue from the nearest bulk water molecules. In our analysis, we define bulk water as 4 or more water molecules within a 4.2 angstrom radius. So to calculate it, we first of all calculate residue depth for all residues in the model protein structure. We then penalize each monolinked residue by its residue depth, and then we sum all of those scores in a model structure, and then that gives us our mod score for that model. We then go on to combine our mod score with our previously defined M and XL score. So this score is for scoring crosslinks, and this flowchart shows an outline of the process. So in this first step, as previously mentioned, we score monolinks using mods, and then we go on to look at the crosslinks. So we will basically give the crosslink a negative flat penalty if it is either inaccessible or if the crosslink itself is too long. If the crosslink passes both of these tests, then we will score it positively based on a probability distribution of different crosslink lengths. That gives us our M and XL term, and then we can normalize both the M and XL and mod scores by converting them to z-scores and add them together, and that gives us our crosslink monolink score, or XLMO. So that gives us this pipeline for scoring performance. So we can, first of all, get our crosslinks and monolinks, and then we will score a benchmark containing a target structure, and then which has 300 suboptimal decoy model structures. So these will range from low C-alpha-RMSE, so very similar to the target structure, to very high C-alpha-RMSE, so a very poor model of the target structure. And then we'll score those using M and XL mods and XLMO. We will then assess the relationship between the score and the model similarity in terms of C-alpha-RMSD, and then we'll calculate statistics. So we'll calculate area under the receiver operator curve, Spearman correlation coefficient, and precision. So because monolinks are not widely used in the field, they are also not widely reported. Therefore, we had limited data to actually operate with. For that reason, we split our analysis into two data sets. So first of all, we had our simulated data set. This contains 200 PDB structures, each of which have 300 suboptimal models. And this was from the 3D robot data set from the Zhang lab. Onto this simulated data set, we simulated crosslinks and monolinks onto the target structures. For the experimental data set, we had some experimental data. So this contains 6 PDB structures, each of which had experimental data, which we reprocessed from the PRIDE proteomics database. So to simulate crosslinks and monolinks, we first of all simulate all possible crosslinks and monolinks on the target structure. We then select a random 50% of monolinks and a random 20% of crosslinks, giving us a realistic experimental recovery. We then randomly resample this 1,000 times, giving us a representative sample of the protein surface. And then we recalculate the scores each time and we take an average. So these density plots show the statistics over our simulated benchmark. So first of all, we can see that MNXL marginally outperforms MODS over all 3 statistical measures. However, when we combine our MNXL and MOD scores in our XLMO score, we see a small but significant increase in performance. That suggests these 2 scores are complementary and that using all this information can be useful in the modeling of protein structures. So as mentioned previously, our experimental benchmark is only 6 proteins large. So we can see from this bar chart the mean XLMO performance is marginally higher than that for MNXL and MODS. However, it's very variable and it's not statistically significant. So the real value of the experimental benchmark is looking at the individual examples in it. So looking at these results in terms of correlation, what we can see is that although XLMO is only the top score in 2 out of the 6 models, it is also never the lowest score. So it always appears to compensate for poor performance of either MODS or MNXL. So let's take human serum albumin as an example. We can see that MODS performs significantly better than MNXL, and XLMO performance is slightly lower than MODS, but much higher than MNXL. Now, it's easy to see from this example that MODS is the best score for this. However, we only know that because we know the ground truth. We know what the human serum albumin structure looks like. When we don't know and we're modeling from scratch, using this XLMO score that includes all of the data from our XLMS experiment is likely to be the best way to score our protein structures using all of the information that we have from our XLMS dataset. So to summarize, monolinks using our MOD score can be used to score model protein structures. And combining the MNXL score for crosslinks with our MOD score for monolinks in our XLMO score can lead to greater performance as shown in our simulated dataset. In our experimental dataset, we see similar performance between the 2 scores. However, we do see signs that there is some compensatory effect between the 2 scores. And if you'd like to know more about this, please see our paper that's been recently published in Structure. Finally, I just want to plug our XLM tools package. It's a Python package which you can use to score model protein structures using MNXL, MODS, and XLMO, and it's available on the TopFlab GitHub. Finally, I'd just like to thank everybody in the Top and Thalassanos labs, everybody involved in our paper, and Birkbeck for funding my research. And thank you very much to you for listening.